I'll just start and then get up to pose one or two questions to the guys up there. And Giorgio, you got a very nice presentation about how principal cation exchange resins differ. So my question to you would be, what resin characteristics do you think will need to be considered or required to meet future manufacturing challenges?
That's a loaded question. Because I think it depends obviously on the application. Generally speaking, I think there is a need for higher resolution resins. If you look at current biopharmaceuticals, the degree of micro-heterogeneity is huge. All kinds of glycoforms, charge variants are present. I think eventually we will move more toward really single molecule species, which then will require the separation of these variants. Currently, that's not achievable with the available resin. So I think maybe 10 years from now, we'll have moved toward HPLC for protein purification, where we can actually resolve charge variants, glycoforms, and so on. So maybe the functional groups will remain the same, but we'll have resins that are able to withstand higher pressure, so using smaller particle sizes for faster kinetics. And that's true whether it's cation exchangers or anion exchangers. I think we will see this trend. So,
where do you think where science would concentrate? So would it more be on the chemical sides of the bead to make it smaller, more pressure stable, or?
Yeah, I think that the base matrix engineering will become critical, because you're still going to need to have large pore sizes, and of course with smaller bead sizes you're going to have higher pressure. So focusing on stronger backbone materials that can withstand pressure would be highly desirable.
Okay. And any comments?
Maybe I can add a little bit more from industry perspective that even though it's not achievable to further resolve those different variants, right now through some sort of high throughput screening to find the resin to achieve much better resolution. Yeah, I definitely see that being a trend. For us, I think it's always a dream for all of us to have such a good resolution to come up with a single species. Yeah.
Yeah, thank you. And any opinion in the audience on that?
In the same line of thought, where we're looking at the properties of the different resins, porosity, particle size, a lot of early stage development, we're doing a high throughput resin screening where we're using automated systems or a TCAN, if you will, to test various different resins, sometimes as many as 30, 60, or a full 96 well plate. And so one of the questions I have is, because of these new advances in resin, should we be bucketing our screens not just on the chemistry that's being used for the separation, whether you're talking about cation exchange or sulfate versus a CM resin, but also on the particle size, the porosity, and any other different properties of the resin?
To answer your question, from the resin development side, we really want to, I agree with Giorgio, that it's application dependent. Depends on whether you are using this resin for the early stage, like capturing your targets, versus the polishing purification, you are dealing with very similar species, either it's product related or from the process. So the advice I usually give customers, first you set out your goal, like how much material you want to process in which time frame. Because we talk to different kind of customers. Some want larger beads because I want to run faster. Some really want small. At the beginning, that I am looking for resolution. But at the end, when they pack a large column, when they see the pressure, they cannot really run fast. They come back to us and like, can we have larger beads again? So there are different kind of solutions for this. Because on one hand, like we have smaller particles, we can have higher resolution, but do have to deal with high pressure. So from our side, we are also trying to generate a narrower cutoff of the particle size, for example, so you can handle the pressure. And from the engineering side, we also look into, for different kinds of resin, whether the packing, the way you pack it, or the dimension of the column, whether it helps you to mitigate the pressure issue. And then for the earlier capturing, also depends on whether you simply want to do fractionation or you really want to have a purified species within one step. So if you do want to look into the distribution of particle size, how that affects your purification, we can talk further so we can actually give you more specific advice. And also we look for feedback from you, like what works for you, then we can really tailor the resin.
I have a question about purification resins. See, you are developing resins for better purification, faster purification, higher efficiency. But for biosimilars, you have to match the originator. And what do you do there? What do you do for biosimilars?
Yeah, it's definitely a new challenge for us. You know, before I joined BI, when I work at the MedMill, we only do the new molecule. So what comes up from my hand, go into the phase 1 clinical trial is what you get, and in the end, you just need to match the quality over there. From what we learned in the past years, developing the new process for the novel molecule for the new target, that I think typically HCP reduction, we have a lot of experience, and aggregate removal, and there's a lot of good choices. To me, what's basically challenging is the fragment removal. I would say right now definitely looking for new technology, resins, talk those areas. And also, if professor had some research work through either model prediction or some understanding of the resin characteristic, a little bit focus on some research to look at those, charge variants would be something very interesting for the industry.
Yes, I think that's a good comment there, because when you're looking at biosimilars, for example, antibody drugs there, the example of charge variants is this type of parameter is usually defined by the upstream process, by a very defined one. And when you're doing a biosimilar, you're having different conditions in the cultivation, in the upstream, and then you have to match your purification, that you meet the charge variant profile of the original drug, and that really is a challenge for the purification there. And and anybody of us who wants to comment on that?
Well, from my perspective, I worked on Remicade, and now you have biosimilars coming out. Well, when Remicade was created in the early 90s, a lot of the technology and analytical tools that are available now weren't available. So you're comparing a biosimilar to something that you actually probably don't have the data to show that it is truly going to be the same. Probably the biosimilars might be cleaner than some of the original just because of the technology at the time of analytical techniques.
Yep, good point.
I was hoping you would make some comment about sort of the resin architecture, particle size, and porosity with regards to the viral clearance. Because in your very nice talk, you only group the resins by their functionality. But there must be something beyond functionality that affects viral clearance. So maybe you could comment on that.
Yeah, of course. Unfortunately, the data that we have so far doesn't include that. But from literature, you can see, especially the past few months, we saw a couple of papers showing that some viruses, especially with protein A, don't bind with the bead itself. So it's a problem with the product, not the resin themselves. There is binding with, could be, and the theory could be, some of these impurities bind to the product and the virus bind to these impurities. And that caused the problem with when elute with it. So you might take it to a different direction with the problem with the product itself and how to purify it. Also, on other perspective, with real, for example, one of the smallest virus that been tested with monoclonal antibodies, the heterogeneity of the virus production could have an effect on the virus clearance process for chromatography. How pure the virus is, and some data show that a non-specific binding of this virus to the beads could cause problem with what is around it. I think we saw recently, although we don't use highly pure ultracentrifuge virus with chromatography step because you don't have it like nanofiltration step, but it starts now to be used more often to get rid of all these background that might have cause that problem for showing poor clearance.
You mentioned that when you're looking at viral clearance in flow-through, that you get better results for the anion exchange viral clearance there. Georgia, you've been outlining different modes of chromatography, comparing frontal chromatography with bind and elute mode. And in this context, should we reconsider how we develop processes? In my opinion, we have high titers of products there. We only need to bind impurities. We would use less capacity, have smaller column size. Would it be interesting to go more into flow-through purification? So what are your opinion, academic, industry opinion there? Resin developers, I'm curious.
Okay. So the academic will tell you that you capture first, because this, I think, helps a lot removing proteases, isolating the product from the very complex mixture. So I doubt that we can have a completely flow-through system. But on the other hand, after the initial capture, then flow-through could be certainly considered for all the remaining steps. Now, except, of course, where you have to resolve product-related variants, because then this is just not feasible to remove dimers in a flow-through mode. I mean, it's really idealistic that we can have the monomer flow-through and the dimer being retained. Okay, maybe in a small-scale system you can do it, but it would not be robust. But
maybe with hydroxyapatite.
Maybe with hydroxyapatite, but that's kind of a magic resin, can do anything. Enterix data were quite interesting, comparing the viral clearance and the flow-through versus the bind and elute. I thought that was really quite nice observation.
Yeah, you can see it also with a mixed mode. The result, it's a flow-through, and every time it's showing much better result in regard to virus clearance. So there's something there that push you to this mode of clearance.
Thinking about the flow-through concept, it kind of lends itself to more continuous purification, which is also kind of a new trend that everyone's been talking about. So I was wondering if there's any resin properties you haven't discussed yet that would lend itself also to continuous processing.
So flow-through definitely offers significant advantage when you want to connect the rest of the polishing step and make it continuous. However, the product quality always the most important consideration. So typically that in the process development, we always try binding the elute first. And from there, and we will look at, maybe we can do flow-through, achieve a similar quality. If not, at least we have a bind elute condition. But somewhere in between, the weak partitioning flow-through is another thing we can consider. That typically works quite well if you have a bind elute conditions figured out. And I personally don't have any failure or any challenging that moving that was to weak partitioning flow-through mode.
For the flow-through versus bind and elute, I think, the virus, there's no question, is going to bind almost exclusively on the outside surface of the beads. So if you're in bind and elute, your product will stick to the surface, which may really reduce the ability of the resin to remove the virus. Whereas if you change the composition of the buffer system so that only the virus binds and the product flow through, you have a much better chance of capturing the virus on the bead surface. I think that's why you see that in most cases the flow through is better for viral clearance.
Yeah, any other comments on that? Georgie, I've also got a question to you again there. You've been outlining frontal chromatography as one mode of operation to consider there next to bind elute there. I'm aware of other concepts like displacement chromatography. Do you know that these types of chromatographies have a future? Because when I look at the industry, everybody develops like mostly in bind and elute. You have a flow-through step for impurity removal. What's your opinion on that?
Yeah, the frontal chromatography is, okay, it's been around in principle for a very long time, but it's only recently that we have seen some applications from Genentech, for example. And maybe it's kind of a niche technique for this dimer removal because obviously it only works when you have a two-component separation, so as a final polishing step, it can be extremely effective. It's isocratic, it's extremely high capacity, and I think it's very robust also. Displacement is another matter. I think we're still limited by the availability of displacers that are compatible with the process. And of course, in all these cases, you need to have fast kinetics because otherwise these fronts become too shallow and you end up with very poor yield. So having the proper resin with large pore size, small bead size, can make these other techniques perhaps applicable for a broader range of separations.
What advice would you give for screening new resins, for example, for platform and non-platform molecules? So what was your perspective as a purification scientist?
Okay. As a scientist, I want to tackle all new problems. So I think, like for me, regardless the platform, the only platform I've seen successful is the protein A for monoclonal antibodies. For other recombinant proteins, anyway, iMac that a lot of us don't like to use iMac for drug production. So for other recombinant proteins, we don't have a platform, but you've seen many cases today, in my cases and also from other speakers. Namely, for protein purification, there are two basics. One is the purification pH. One is the buffer conductivity. So mostly we start from the simple conditions. And if I start developing a method for unknown protein, I would first look at the stability of the protein, like whether it's stable under extreme pH. Is it going to be precipitating if I use high salt or low salt? So you use this kind of information to define your screening window, because these days we are not limiting, because we have the TCAN, we have spin column, we have the atoll pipette columns. You can screen a lot of conditions, but knowing the stability of your protein will direct you to the right operation window, so you won't have to at the end, like when you try to scale up, you get your surprise. And the second thing, I have been talking about this for a while, is like most of the time when you get the literature from vendors, you see the comparison of binding capacities, that my binding capacity is 300 mg per mil versus 30 mg per mil. But this is something that we as vendor choose the best condition for my poster child protein to show you the binding capacity. You are not dealing with a purified protein. You are actually dealing with a mixture that you have thousands of impurities that you don't know what they are. And also you are dealing with proteases in your system that at the end of the purification, you lose your material. You don't know where the fragments went to, for example. And also another important consideration is the recovery of the target, either by mass and by activity. So I would suggest that this should be put into consideration at the beginning of your screening. That you do more work at the beginning, it will help you a lot down the street. And another advice, I've been promoting mixed mode chromatography. For advice for that is like a lot of people hesitate to screen mixed mode, failing that I cannot use my PI, predicted PI, to start a purification. But in reality, screening a mixed mode resin is the same as screening any other resins. It's like you look at the effect of buffer pH and salt concentration. So you first get the first set of preliminary data, so you can see the distribution of the target versus the impurities. So based on that, depends on whether it's the capture step or it's the polishing. So you decide whether you want to have the target bound versus have the target in the flow through. But that actually, from the first screening, the preliminary data you already tell, although you don't know what's the interaction mode to start with, but with the condition, the behavior of the target, you know what kind of interaction you are dealing with. And from that on, you can refine the condition. So the lesson is like, don't afraid and just try it.
Okay.
Yeah, so that is good strategy, but for someone like, I'm always very practical. If I have a 10 project a year, and each project has a very defined timeline, that I was always thinking about, for any non-mAb, can we group them into different molecular format and try to design the platform for each of them? The selection of the resin for each type of molecule can be done either by design. For example, I really like Xiumei's presentation that the Nuvia Q resins with a pore size looks like really work very well for vaccine. So that can be the good choice of the platform for that type of the molecule. And one at a time, so either by design or either by screening or either through the collaboration with the resin company, that we can start establishing the platform for other non-mAb and one type at a time. So that's always, I feel, more practical for industry, especially this day, the development time is not that much. Every project talking about acceleration. And another approach is affinity through the high throughput screening of the ligand, and make the capture always as affinity chromatography. And I think that's also another valid option for the non-mAb.
Would you consider like multimodal interactions, which we probably consider as pseudo-affinity, a valid tool for non-mAb purification platforms?
I feel that, one-size-fits-all approach may not work very well for all the molecules. But however, one-size-fits-all within a certain group of the molecule, that's probably still doable. So mixed mode, to me, by design, is more like one-size-fits-all can do everything. But then what is good about it? What's specifically good about certain molecules that you still need to do a lot of the optimization. And in the end, I still don't have the confidence within, for example, 2 months, 3 months. Do I get my, process? And I'm not quite sure. But however, if I go to affinity through a high throughput screening of the ligand within 3 months, I'm pretty sure I can find something more specific than probably mixed mode.
Okay, good. Yeah, so Xiumei, comment on mixed mode. It's your topic, I guess.
Yeah, since we screen a lot of structures in the lab, so to adjust this, this request, I would say like we usually, like if you have a specific request, we work with you. Because mostly if we try to compare the capture with protein A versus all those ion exchanger, the very specific difference is like the conductivity of the buffer, like whether I can tolerate salt or not. Because when you grow the culture, if the target protein is secreted to the media, you want to be able to capture it directly. So if we estimate that conductivity, like it's around 100 to 150 molar sodium chloride. So in terms of salt conductivity or salt tolerance, if we can develop a resin at this time, you can have the target bound. And after it's bound, you can recover at least 90% of the target. That will be pretty equivalent to a protein A kind of affinity. Although we are not talking about a specific interaction between the ligand with a couple of residues. But if we can find the ligand for you, that the binding happens between 100 to 300 millimolar sodium chloride, then you have a much larger role. Depends on the interaction mode. You can either go higher pH, lower salt. If it's a hydrophobic interaction. If it's a charged interaction, we go even higher salt to have it eluted. So we are gaining more experience by looking at different ligand structure and how to link this ligand onto the base beads to fine-tune the selectivity. But the first goal I would set as tolerance to modest concentration of salt. That's like affinity.
I also want to thank everybody here in the panel again and you in the audience for taking your time and contributing there. Thank you very much again.